Hello everyone, I'm Apoorva Shankar from the Department of Life Sciences, Christo Jayanti College. Today under foodborne infections and intoxications, let us discuss about what are foodborne infections and intoxications. Introduction. The foodborne diseases may be caused by bacteria, bacterial toxins, viruses, fungi, fungal toxins, zoonotic parasites, protozoans, pesticide residues, drug residues, heavy metals, food adulterants, and food additives. The food poisoning can be of three different types broadly, that is, foodborne infection, foodborne intoxication, and toxico-infection. The foodborne infection is caused by the ingestion of viable pathogens along with the food. For example, that is typhoid. Foodborne intoxication or food poisoning is caused by the ingestion of the foods that is containing preformed toxins like botulinum, staphylococcal poisoning. Toxico-infection, that is the organism that produce toxins in situ when ingested along with the food, is called toxico-infection. Example, Bacillus cereus infection or the Bacillus cereus poisoning. The foodborne diseases can be broadly divided into food poisonings and infection. The poisonings can be chemical poisonings and intoxications. The intoxication can be because of the poisonous plant tissues, poisonous animal tissues, and microbial intoxications. Under microbial intoxication, we have three different types, which is caused by algal toxins, mycotoxins, and bacterial toxins. Under bacterial toxins, we have different types of toxins, that is, enterotoxins and neurotoxins, and the others which interfere with the carbohydrate metabolism. And under infections, we have enterotoxinogenic infection and invasive infection. Enterotoxigenic infection can be because of the sporulation, growth, and lysis. The invasive infection can attack the intestinal mucosa, it can be systemic, or it can affect other tissues like the muscle and liver. This is a broad classification of the foodborne diseases. The foodborne diseases are the figure examples of the bacteria that is responsible for the foodborne intoxications and infections. So intoxications, we have staphylococcal intoxications, which produces an enterotoxins, which is produced by Staphylococcus aureus, and botulism, which is a neurotoxin produced by Clostridium botulinum. Infections such as salmonellosis, which is caused by enterotoxin and cytotoxin produced by Salmonella species. Example of infections include Clostridium perfringens, which causes illness, Bacillus cereus, which causes gastroenteritis, enteropathogenic Escherichia coli infections, and others like Yersiniosis, Shigellosis, Vibrio infections are caused by the bacteria. The differences between what is an exotoxin and an endotoxin. The exotoxins are proteins which are produced inside the pathogenic bacteria, most commonly gram-positive bacteria, as a part of their growth and metabolism. The exotoxins are then secreted or released into the surrounding medium following the lysis of the bacteria. Whereas the endotoxins are the lipid portions of the lipopolysaccharide layer, which forms the integral part of the outer membrane of the cell wall of gram-negative bacteria. So this lipid A serves as an endotoxin. The endotoxins are liberated when the bacteria die and the cell wall breaks apart. Foodborne infections. The different types of foodborne infections, the mycotic foodborne infections include the Candida, Sporothrix, Vangilla species, etc. Viral foodborne infections include hepatitis A, E, and Norwalk virus, poliomyelitis virus. Parasitic foodborne infections includes hytidiosis, teniasis, trichinosis. Protozoal foodborne infections include cryptosporiosis, toxoplasmosis, sarcocystosis, and cyclosporosis. Let us take an example of Clostridium. Two species of Clostridium are of significant importance in food industry, namely the Clostridium perfringens and Clostridium botulinum. Clostridium perfringens, which is gram-positive rod-shaped bacterium, which is capable of forming end and dormant spores, can change potentially harmful vegetative cells if exposed to the cooking temperatures and when allowed to stand at temperatures between 41 to 120 degree Fahrenheit. The vegetative cells are killed when the foods are cooked at or above 140 degree Fahrenheit. However, the spores can survive the process of cooking. It is an anaerobic organism and hence can thrive in very little or no oxygen conditions. It doesn't grow at refrigeration or room temperature. So, this point suggests that this organism is a common contaminant in the canned foods and vegetables. The organism is found abundantly in soil, water, sediments, and the intestine of domestic animals and human sewage areas and which are prone to sewage contamination. They can cause food poisoning when people consume large numbers of this bacteria, which they can multiply and produce toxins inside the intestine. When people eat contaminated food with toxin already present in it. The common sources of Clostridium perfringens infection include the root vegetables, canned foods, meat, poultry, gravies, and other foods which are cooked in large batches and held at an unsafe temperature. Outbreaks tend to happen in places that serve large groups of people, such as hospitals, schools, cafeterias, prisons, nursing homes, and other events with catered food. The disease cycle, the symptoms are caused by a toxin that is produced by the bacteria. When the large numbers of vegetative Clostridium perfringens bacteria are ingested, bacteria that survive the acid conditions in the gut grow in the intestines, form into spores, and release an enterotoxin that results in diarrhea. When Clostridium perfringens is introduced into deep wounds, the toxin produced results in the damage to the skin and deeper tissues. Symptoms: most people with Clostridium perfringens infection develop diarrhea and stomach cramps within 6 to 24 hours after eating contaminated food. The illness usually begins suddenly and lasts for less than 24 hours. Diarrhea can cause dehydration, so it's important to drink plenty of fluids. This infection usually does not cause fever or vomiting, and it cannot be passed from one person to another. The diagnosis of this Clostridium bacterium is usually done with the stool samples, which are analyzed for the presence of the pathogen or the enterotoxin. The prevention, cooking in right temperatures, helps to destroy this bacteria as this bacterium is sensitive to cooking. Prompt refrigerations and leftover cooked meat and reheat it thoroughly. The internal temperatures before serving. Proper storage of canned foods is important. Treatment: the food poisoning from Clostridium perfringens is mainly supportive as it will resolve on its own. Clostridium botulinum. Of many types of foodborne illness, botulism is one of the most dangerous, and it can cause paralysis and it can be life threatening. But it is rather rare. Botulism is caused by the bacterium Clostridium botulinum. It releases a neurotoxin, which is a poison that attacks our nervous system. Clostridium botulinum is an anaerobic rod-shaped spore-forming bacterium that produces a protein with a characteristic neurotoxicity. Under certain conditions, these organisms may grow in foods producing toxins. Botulism is a severe form of food poisoning, results when toxin containing foods are ingested. The organism is resistant to heat, radiation, drying, and the spores can survive several hours of boiling while toxins are heat sensitive. Seven types are distinguished on the basis of serological specificities of their toxins. Type A is the one commonly found in human botulism and is more toxic than the type B. Type B is found often than type A in moist soils and is less toxic to humans. Type C causes botulism of fowls, cattle, and other animals but is not reported in humans. Type D is associated with the forage spoiling of cattle. Type E is toxic to humans and has been reported from the fish and sea products. Type F produces human botulism. Type G has been isolated from the soil but not reported botulism in human. The Clostridium botulinum is widely distributed in soils and in sediments of oceans and lakes. The findings of the type E in aquatic environments by many investigators correlates with the cases of type E botulism that were traced to contaminated fish and other seafood. Type A and type B most commonly encountered in the foods associated with the soil contamination. Here, the symptoms include double and blurred visions, drooping eyelids, difficulty in swallowing, slurred speech, and shortness of breath and weakness. These are mainly caused because of the neurotoxin. The diagnosis can be done by the stool samples, which are analyzed for the presence of the pathogen or toxin. The prevention, the botulism toxin can be killed at high temperature. So if you're heating home canned food, consider boiling it to 10 minutes to kill the bacteria. Proper refrigeration can also help to prevent the growth of Clostridium botulinum. Treatment is through antibiotics and antitoxins. The contamination in the canned food can be indicated by the bulge or the container spurts out foam or liquid when it is open, and the content smell unusual or foul smelling can be observed. So this is one of the reports where in 2017 September, the foodborne botulism, a nacho cheese I had only nacho cheese is a case that is reported with the foodborne botulism. Here, a 32-year-old female presented an emergency department with complaints of diplopia followed by dyspnea, chest tightness, and congestion. So this is the one of the case reports of botulism which was observed after eating nachos cheese. That was about foodborne infection, intoxication, and clostridium. Thank you.